Welcome. Today we're going to deconstruct a GCSE biology exam question together. We'll look at enzymes, we'll look at plants, and we're going to use the official mark scheme as our blueprint to build the perfect answers.
So here's our game plan for today. We'll break down question 04 into 5 manageable parts, tackling everything from the lock and key theory of enzymes all the way to interpreting experimental data.
Okay, let's dive right in. You know, question 04 is a really great example of how exams love to mix things up. It tests your knowledge on 2 super important areas, how enzymes work and a classic investigation into food production in plants.
First up, the question gets going with lipases. Now, these are the enzymes that break down lipids, fats. It starts with a 2-mark question asking what lipids are made of. Let's see exactly what the mark scheme demands for full marks here.
All right, now let's get into the core theory. This next bit is all about enzymes, specifically how they work and why they're so incredibly specific. Getting this down is absolutely fundamental to acing this question.
And there you have it. For those first 2 marks, the mark scheme is crystal clear. The 2 products are fatty acids and glycerol. You've got to name both of them to get the points. Simple as that.
Okay, so next up, they give us this classic diagram and ask us to explain the lock and key theory using what we see. So the big question is, how do we turn this simple picture into a perfect 3-mark answer?
This is exactly what the examiners are looking for. It breaks down into 3 key steps. Step 1, the enzyme and the substrate bind because their shapes are complementary. That's a keyword. Step 2, the substrate gets broken down into products. And step 3, those products are released and the enzyme itself isn't changed. It's ready to go again. So, what's the crucial point for the next part? It's all about one word: specificity. Why does this lipase only work on lipids? Well, the mark scheme spells it out for us. Each active site has a specific shape. It's so unique that it only fits one type of substrate. Just like a key only fits one lock.
Right, the question is about to switch gears now, moving from pure theory to the practical side of things. We need to describe the chemical tests that are used to identify both glucose and starch. This is all about recalling those standard lab procedures.
Now listen up, because to score all 3 marks for the glucose test, you have to hit these 3 points, no exceptions. First, you add Benedict's solution. Second, you have to heat it. And third, you describe that positive color change, from blue all the way to green, yellow, orange, or even brick red. Miss one of those, and you'll miss a mark.
And for the starch test, it's a bit easier, just a 2-mark process. According to the mark scheme, you get 1 mark for saying you add iodine solution. And you get the 2nd mark for describing that classic color change from its normal yellowy brown to a deep blue-black if starch is there.
All right, so we've got the theory down, we know the method, now comes the fun part. We get to play scientist and figure out what this data is actually telling us. This is all about explaining the why behind the results. Here are the results the students recorded, all laid out in this table. Now, take a good look at the differences. The leaf that was kept in the light has a strong positive result for glucose, and it's positive for starch. But the one that was moved into the dark, it only has a weak positive for glucose and a negative result for starch. So what's going on here?
And here's how the mark scheme breaks it all down. For the leaf in the light, think of it as a busy factory. It's making glucose through photosynthesis, and any extra glucose gets converted into starch for storage. But the leaf in the dark, it's in survival mode. It's breaking down its starch stores back into glucose to use for respiration, because with no light, it can't make any new glucose.
And for the grand finale, the last part of question 04 asks you to put on your scientist hat. For just 1 mark, you need to suggest a way this investigation could be developed to find out even more. This really tests your ability to think critically. And you know what? For this last mark, the mark scheme gives you a ton of options. There isn't just one right answer here. You could suggest testing different parts of the plant, like the roots or stems. You could test other species of plants entirely. You could get more precise and actually measure the concentrations, or you could play around with the time the plant is in the light or dark, or even use variegative leaves. Any one of these is a perfectly good answer.
So, what's the big takeaway from all this? Well, we've seen how enzymes have that specific shape, how plants make glucose through photosynthesis, and how they cleverly store it as starch. But the real lesson here is learning how to use the mark scheme as your guide. Now that you know how to use it as a blueprint, just imagine what other exam questions you could deconstruct to build your perfect answer.